जी अस्सलाम वालेकुम. Can you people hear me?
Yes.
अच्छा जी, एक तो आप बेटा अपने ओरिजिनल नाम से ज्वाइन इन करें. अगर तो आपने किसी और अकाउंट से ज्वाइन किया हुआ है तो आप अपना नाम चेंज करें, क्योंकि इसी से जिस नाम से आप एंटर होंगे उसी नाम से फिर आपकी अटेंडेंस जो है वो सेव होगी. ओके. और इफ यू हैव ज्वाइंड विद डिफरेंट नेम देन योर अटेंडेंस विल बी मार्क्ड एब्सेंट ऑब्वियसली. सो आप रीज्वाइन कर लें इफ यू आर विद द डिफरेंट नेम. सो हमने सेल मेम्ब्रेन खत्म कर लिया था. आज हमने एंजाइम शुरू करना है बेटा.
Yes, sir. Sir, I believe we still have to do endocytosis and exocytosis. We haven't done that. Movement in and out of the cells. वो हमने पूरा complete नहीं किया अभी.
ठीक है. एंडोसाइटोसिस एक्सोसाइटोसिस हमने सेल में जब लाइसोसोम का रोल किया था तब भी कर दिया था गोल्गी बॉडीज फॉर्मेशन के साथ ही. You want to do that again, we can do that again. However, we have done that with the cells when we discussed the role of Golgi bodies and the role of lysosomes. So we discussed both endocytosis and exocytosis. However, so let's see enzymes today. Okay, so in this chapter, we'll discuss properties of enzymes, as they are globular protein and they catalyze the chemical reactions. So there are two types of enzymes. We'll discuss two types of enzymes, intracellular and extracellular enzymes. Then we'll discuss model of enzyme action. Okay, so today we'll discuss these two things. Then we'll continue with the progress of an enzyme catalyzed reaction, the use of colorimeter, the effect of the different factors on the rate of enzyme action, and then finally immobilization of enzyme. So these are the topics included in this chapter. Okay, so we will need three more lessons to complete this chapter. So let's see. Enzymes can be defined as or described as biocatalysts. So what does this biocatalyst mean? Can you people hear me? Okay, so as they are biological molecules, they are globular proteins, and they speed up a rate of a chemical reaction. So there are many things that could speed up the rate of a chemical reaction. Temperature can increase the rate of a chemical reaction. Substrate concentration can increase the rate of a chemical reaction. Okay. So how these enzymes then differ from the other factors which can speed up the rate of a enzyme action? These differ from the other factors in such a way that they are not used up during a chemical reaction. Instead, they remain unchanged at the end of a reaction. If we look at the other properties of enzymes, other than properties of enzymes, so as we said earlier, they are globular proteins. So this is number 1 point. Then they speed up the rate of a chemical reaction without being changed at the end. This is another property of an enzyme. Then enzymes lower down activation energy. There are some more points that we can add up with globular proteins. They are water soluble. Okay. And they have a 3D tertiary structure. So they have a single polypeptide generally. And they're specific to pH, temperature, and substrate concentration. Okay. So let's see what is an activation energy. Activation energy. EA, energy of activation, yeah, AE, both the ways we can write, energy of activation or activation energy. Every chemical substance that has some chemical potential energy, if on this y-axis we plot energy of different chemical substances. So if this is, for example, a glucose or any other substrate, it has some specific chemical potential energy. If glucose is broken down to carbon dioxide and water, they do have some chemical potential energy, which is lower than that of a glucose. So if this is a substrate and these are the products, if substrate has higher chemical potential energy and the products have a lower chemical potential energy, then there will be a net change of energy, which is known as an enthalpy change, or which is known as minus delta E. So this is a change in energy between the substrate and product. So this would be an exothermic reaction. During this reaction, energy is released. Okay. So substrate should convert into the products at its own, but it doesn't happen so. You see, you have glucose or a cellulose in your paper, in your books, and there is oxygen in the atmosphere. But oxygen and glucose, or oxygen and these carbohydrates, don't react at their own, and they don't convert into the product. To convert these chemical substances into the product, there is some amount of energy needed at the start of a chemical reaction. For example, if you burn a small piece of a paper or side of a paper, you will see this burning start at one end of a paper, and then it continues throughout the whole paper, and then maybe the other papers that are lying around. So you see, you started with providing a small amount of heat, which started this chemical reaction. It's the same that you do with burning the matchstick. You burn the matchstick with the that chemical that is present on the side of a match. So this friction actually produces or releases uh, you know this releases that energy needed to start that burning of that matchstick. So this initially uh, a higher amount of energy needed to start a chemical reaction is known as an activation energy, and that could be plotted on a graph paper, on a graph this way. This hump made in this graph that shows an activation energy. This is initially higher amount of energy needed to start this chemical reaction and is known as an activation energy. This is without an enzyme. Okay. When enzymes are not used, a higher amount of activation energy is needed. But when enzymes are used during a chemical reaction, even that chemical reaction can take place at a lower chemical potential energy. For example, you already know that if this sucrose is boiled with dilute HCl, it will break down into glucose and fructose. So this boiling provides that activation energy needed to break down this sucrose into its products. Okay, so this reaction can be catalyzed even without an enzyme. So this reaction is without an enzyme. However, we can catalyze this reaction in the presence of an enzyme, sucrase, and we can break down this sucrose into its products, sucrose plus H2O, and you use an enzyme sucrase, which will break it down to glucose plus fructose. And this reaction can be catalyzed at any lower temperature like 20 degrees Celsius, 30 degrees Celsius, and even it will be faster than the one that you carry out without an enzyme. So you see, these chemical reactions can be carried out with an enzyme or without an enzyme. But if this enzyme action has to be carried out without an enzyme, you need a higher energy and extreme conditions to be provided so that this substrate can be converted into the product. Enzymes make even these chemical reactions possible at lower temperature. Okay, so still they need some energy at the start of a reaction, which is known as an activation energy. So if this reaction is catalyzed uh, in the presence of an enzyme, then this energy will be lowered down, and the new activation energy with an enzyme will be, this is an activation energy with an enzyme. Okay, and here do you see this is a difference or decrease in energy, activation energy, without an enzyme and with an enzyme. So this is difference in an activation energy. Okay, and then let's see, we have some questions on this activation energy. So let's see these questions. So let's see, the graph shows energy changes in a chemical reaction. What is the activation energy when an enzyme is added? So you see this is higher activation energy. So this is this energy from here to here. This is without an enzyme. So this is activation energy without an enzyme. Okay, and this is the lower activation energy. So this is with enzyme. Okay, and here this difference is actually without and with an enzyme. So what is the activation energy when an enzyme is added? So it should be 2 only. Okay, the graph shows the effect of an enzyme on a reaction. Which labels are correct? Look at this question. Can you answer this question? Fatma, can you answer this?
There, C,
Yes, C is a correct answer. Why not A?
because W is the difference between the activation energy and X has a high activation. W is basically the difference between two activation energy.
So W is not the activation energy. W is the difference in the activation energy. Okay. So that's why A is not a correct choice. Why B is not a correct choice?
Z is basically the difference of Z is basically the enthalpy change.
X is the reaction with the enzyme. No, X is not. Z is the overall energy change. That's fine. That's okay. Second part is okay. But first part is X is the reaction with the enzyme. So X is without the enzyme. So the first part of this statement is not correct. Second part is okay. Y is the reaction with the enzyme. Yes. W is the difference in the activation energy. That yes, both the parts are correct. And why D is not correct? Z is the energy gained by the product. No, it is the energy lost by the substrate. W is the activation energy with the enzyme. No, it is the difference. So that's how this is. C is the correct choice. Great. So explain how enzymes lower the activation energy needed to uh, allow reactions to produce. We will discuss this in lock and key model. So let's see. The next topic is lock and key or model of an enzyme action. Model of an enzyme action. Okay. There are two theories proposed to explain the mode of an enzyme action. One is lock and key model or hypothesis, and the other is induced fit model. Okay. Or maybe the induced fit hypothesis. According to lock and key model, an enzyme has a 3D rigid shape. So this is an enzyme. Okay. So this enzyme has a rigid shape. It is obviously 3D globular shape. It is water soluble, that we have already discussed, and it is a tertiary structure. But the shape of an enzyme is rigid. It doesn't change its shape. This is an active site. Active site. And the active site has complementary shape to the substrate. So if this is, for example, a substrate, this is a substrate molecule. So shape of an active site is complementary to the shape of a substrate. Okay. So when enzymes are colliding, enzyme and substrate molecules are colliding with each other due to the kinetic energy in a reaction mixture, they both hit with each other or collide with each other, and a successful collision may take place between a substrate and an enzyme's active site, so that a substrate binds into the active site so this is exactly the same. I cannot make that ditto copy again, but you should understand that it's the same. Or let me just copy it from here so that it is exactly the same. Okay. Or let me just copy this whole. Okay, so you see, this is substrate, and substrate and enzyme active site have a complementary shape. So this substrate binds itself into the active site. So it binds here. So it's precisely, uh, complementary, though I have not made it precisely complementary, but they are precisely complementary with each other. And they bind into the active site and held in the active site by the hydrogen bonding between the active site and substrate. Okay. And this makes an enzyme substrate complex. This is ES complex made. Enzyme substrate complex. Now this enzyme, as it makes hydrogen bonding with the substrate, so this enzyme may puts strain at some bonding in a substrate molecule, or it reorients the bonding in a substrate molecule, or brings the substrate molecules together so that a substrate is converted into the products and enzyme remains unchanged. Okay, so enzyme is unchanged, that remains as such, but the substrate is converted into the product. So this may be converted into products. So enzyme is unchanged at the end of a reaction. So you see, the substrate and enzyme's active site are complementary in such a way that a key is complementary to a lock. So the substrate acts as a key and an enzyme acts as a lock. So this is why it is called lock and key model. Hope you have understood it. Then let's see induced fit model. What is an induced fit model? According to induced fit model, this is, for example, an enzyme. And this is substrate. And this is an enzyme. So do you see the active site? This is an active site. Active site. So do you see active site is not complementary? Active site is not complementary to the substrate. Okay, so active site is not complementary to the substrate. However, this enzyme is also specific to, to a substrate. So a substrate molecule binds with it. Let me just copy it. Okay, and this also. Okay, so let's see. This binds with it. Okay, so this binds with it. And you see the active site is not complementary. And induces the change in the shape of an active site. It induces the change in the shape of an active site so that an enzyme is complementary you see. So when this substrate binds, substrate induces a change in the shape of an enzyme so that active site is complementary to the substrate. And so it becomes an exact fit after it substrate binds into enzyme. So that shows that enzyme is not rigid. Enzyme is flexible in its shape. In a lock and key model, enzyme is rigid. It is not flexible, but in induced fit model, enzyme is flexible. It changes its shape, and that shape is changed by the shape induced by the substrate molecule. And then what happens exactly as enzyme substrate complex is now formed? This is an ES complex. So similarly, enzyme lowers the activation energy, and it reorients the bonding and holds the substrate in an active site with the help of hydrogen bonding. And catalytic R groups or R groups of this enzyme that catalyze this substrate into the product. So substrate is converted into the product, and enzyme returned back to its original shape, to this original shape that it had at the start. Okay. So this enzyme is unchanged at the end of a reaction. Yes, it changes during a reaction. Okay. So it changed the shape during reaction, but at the end, it is unchanged again. So that's how induced fit model that differs from lock and key model. Some enzymes follow induced fit model, and some enzymes follow lock and key model. Enzymes which have fixed shape of the substrate follow lock and key model, but the enzymes which have slightly deviations in the substrate molecules, they follow the induced fit model. So let's see. Explain what is meant by an enzyme. So as I said, enzyme is, a biological catalyst, and it is, biological molecule. It is biological catalyst. It increases the rate or a speed of a chemical reaction, and it increases the rate or speeds up, catalyzes the metabolic or biological reaction. It is globular protein. So when you write protein, it is must to write globular, then only you will get mark. And it remains unchanged at the end of a reaction and is not used up and lowers activation energy. In human tears and saliva, lysozyme acts as an extracellular enzyme. State what is meant by term extracellular. An enzyme which acts outside the cell. Acts outside the cell is extracellular. And intracellular is which acts inside the cell. Okay, so digestive enzymes which are released from the pancreas into the duodenum, or the pepsin which is released into the stomach, these are all extracellular enzymes. They work outside the cell. But enzymes which are involved in conversion of glucose to glycogen or conversion of glycogen to glucose, they carry out this reaction inside the liver cells, so they are known as intracellular enzymes. There are many other enzymes, the enzymes involved in respiration, etc. So let's see this other question. A disaccharide tree hallose is a reserve store of energy for Saccharomyces cerevisiae. This is name of yeast, a fungus. When glycogen stored decreases, the monomer of glycogen and trehalase is alpha glucose. The hydrolysis of trehalose is catalyzed by two different enzymes produced by Saccharomyces cerevisiae, regulatory trehalase and non-regulatory trehalase. So there are two types of these enzymes found in this yeast. Regulatory trehalase is found only in the cytosol. Cytosol is, a fluid part of the cytoplasm. And non-regulatory trehalase has been found on the external surface. Non-regulatory is found on the external surface of the cell and inside the cell. State the location inside the cell where non-regulatory halase is likely to be found and explain the reason for your answer. So where this non-regulatory trehalase is found in the cell. So this is non-regulatory trehalase. It is found inside the cell, but here it gives its function. It causes hydrolysis. Okay. The hydrolysis of trehalose is catalyzed by two. So as it causes hydrolysis, so hydrolysis takes place in a cell by hydrolytic enzymes, and these hydrolytic enzymes are present in lysosomes. So this must be found in lysosomes. Why it is found in the lysosomes? Okay. So obviously it is found in the lysosomes because it is hydrolytic enzyme and pH or conditions needed for hydrolysis are provided by lysosome. So that is why it is found in the lysosomes. Is it okay? Let's see now. Explain whether both types of trehalase, regulatory and non-regulatory, can be described as intracellular enzymes. Obviously, we discussed that regulatory trehalase is found only in the cytosol. So this is found in the cytoplasm. So this is intracellular. And the other one is also found in the cell. So that is also an intracellular. So both of these enzymes are intracellular. Okay. So both work inside the cell. So they are both intracellular. Achaji, this is mode of an action of an enzyme. Elastase has an active site with a specific shape. The mode of action of this enzyme supports the lock and key hypothesis. Okay. Explain the mode of action of elastase. You may use the space below to draw a diagram or diagrams to help your answer. You may draw the diagrams here, but you don't need to. If you're not good at drawing, so you should not try that. You will simply waste your time. You should describe it here. Okay. So how we describe this lock and key model? Let's see. In lock and key model, we discussed that shape of an active site is complementary to the shape of substrate. Okay. So this is reject matching or same. There is difference in complementary and matching. For example, if you have this, maybe, this is a board marker, you have this another board marker. So do they have these two shapes similar, or do they have these two shapes complementary? And you see, we have this, for example, this is a cap, इस बोर्ड मार्कर की कैप है. तो ये जो शेप है आपस में कैप की और इसकी बोर्ड मार्कर की, ये आपस में कॉम्प्लीमेंट्री हैं. और ये जो शेप्स हैं दोनों, जहां पे ये दोनों एक जैसे, ये सेम है. So shape of an active site and the substrate are complementary, not matching or not same. So substrate fits into an active site. Why? Because it is complementary, so it fits into that. Lock is enzyme and key is a substrate. And this results into a formation of enzyme substrate complex. As a result of successful collision between enzyme and substrate binds into an active site and enzyme substrate complexes are formed. Peptide fragments, peptide product released, formed. So what are these peptide fragments, products? So it depends, as this question is about elastase. So elastase is an enzyme which breaks down a protein, elastin. So here this reference to this, its role is given here in this point. Okay. Achha. Now hydrogen temporary bonds formed between enzyme active site and the substrate. So this is how the substrate that binds into an active site, or details of lowering of activation energy, and it puts a strain on the bonds, and there may be an electron transfer that takes place between the substrate atoms, and reactants hold close to form bonding. So these are the different ways when a substrate is bound into an active site, the enzyme converts substrate into the product. Okay. So that's how lock and key model can be explained. Lysozyme uses the induced fit model. This uses induced fit model. Explain the mode of action of an enzyme that uses the induced fit model. So in this case, what we should do is, the shape is not complementary. You see, substrate shape not complementary to the active site. Active site partially flexible, changes shape slightly when substrate binds, and active site and substrate now complementary. So when substrate binds, substrate induces change in a shape of active site, so that now active site is complete exact fit to the substrate. So then enzyme substrate complex is formed, and obviously after this complex is formed, the substrate will be converted into the product by an enzyme by lowering of activation energy, or by interacting with the substrate molecule. We have another question. Figure 4.1 shows two ways in which enzyme interact with their substrate. Enzyme A and a substrate. This is an enzyme substrate complex, ES complex, and this is also NES complex. So explain the difference between the two ways in which enzymes interact with their substrates as shown in figure 4.1. Can you explain the differences in these two. What is this model and what is this model?
Enzyme A has a lock and key model, whereas enzyme B has induced fit.
And what is the reason for that?
Because enzyme is changing its shape, the active site, so that it can be more complementary towards the substrate.
So that's why
Kashi.
it is an induced fit model. And enzyme A already has a complementary shape. There is no change taking place when it makes an enzyme substrate complex. So it is a rigid shape. It is not flexible. A has a rigid shape, B has a flexible shape. Okay, so that is why B is an induced fit model. Okay, so enzyme A is lock and key model. B uses induced fit model. Enzyme work by lock and key and induced fit model. Enzyme A, lock and key, shape of active site is complementary. And enzyme B, induced fit as an active site that molds around the substrate or that changes the shape. Molds around or changes the shape. You can change the wording. Okay, changes the shape. So that's why it is induced fit model. So that's all about it for today. In the next class, we'll discuss tomorrow the factors affecting progress of enzyme catalyzed reactions and factors affecting the rate of enzyme action. Okay. Any question? No? So that's it for now. I'm ending this meeting. See you tomorrow. Allah Hafiz.